So we are continuing our talks on a second topic, which is a challenge, which is about the challenges of establishing HTS in a laboratory. And our first speaker is Sébastien Massart. I was lucky to meet Sébastien in the framework of cost action. And so that was starting the work on high throughput sequencing. So, and it's always been a pleasure to work with you, Sébastien. It's the same for the other people, but Sébastien is outside the NPPO circle. Oh God, I'm digging my grave. Sorry.
I'll remember.
That I'm digging my grave. No, but so I think it's really nice when you can establish collaboration a little bit outside the regular circle of the NPPO official labs. So you're professor of plant pathology at Jean-Blou Agrobiotech University in Liège, and you focus your research on diagnostics. And I think Charlotte mentioned it this morning already. Sébastien has been leading a work package in Valitest that was partly about high throughput sequencing and also on statistical analysis. And so, Sébastien, please.
Thank you, Françoise. And I can say as a bit outside the NPPO world that I understood what is a review correction by EPPO when I start collaborating with Françoise and Charlotte. This is really nicely done and it's a real full review. Even scientists in research don't go to these details, but that's really interesting to learn that. So I will exchange a little bit with you about computer, informatics, not going into details, be cool. First, let's say 20 years ago, we were happy. We were lab. We were very happy with morphology or real-time PCR, like mentioned by Renaud before, and we were thinking, yes, this is the ultimate tool for diagnostic, we will solve the things, that's nice. 10 years ago, we get an earthquake, high-throughput sequencing was coming, and we were all thinking, what the hell I have to do informatics and bioinformatics, I'm not trained for that. I'm either a plant pathologist or molecular biologist, how do I do that? So, yeah, we get a bit, not scared, but wow, like strange things, but somehow, today, I will try to show you that we are at a good moment, a good sign for really adopting it at a wide scale. And you have already some example earlier this morning on the progressive wider adoption of high-throughput sequencing, its progressive use in routine diagnostic, its accreditation, we'll also talk after. So, just to give you an example, a personal example. I started 15 years ago on high-throughput sequencing, and we made a first publication here for amplicon sequencing, easy stuff normally. But at that time, I remember there was 1 month of a bioinformatician and me were exchanging with each other on a super calculation cluster built by the company to carry on a basic amplicon sequencing analysis. And if I look afterwards in 2020, 10 years after, the same kind of analysis could be carried out by a scientist on a personal computer. In fact, usually developed a pipeline that Anne used on its own computer to make the same kind of analysis. So it shows you that we start from big cluster, complex to run, toward personal computer for some application, not for all, we will see. So this progressive matching was driven by 2 main elements. The evolution of computing power, and you're aware of that. There is the Moore's law of the doubling capacity of the computer every 18 months. So your computer is more and more powerful to run the calculation needed for high-throughput sequencing. But also, there is another very important element. This is the algorithms. They have been optimized more and more, and they are now easier to use. They are quicker to analyze. At the beginning, these algorithms were not optimized and were really hard to run with a lot of problems. Now, the most important algorithms are really streamlined products. We do not have to debug them anymore. Because, in fact, bioinformatics is quite important, and I show you here an illustration. In fact, the bioinformatics part, okay. But here you have the lab part in blue, and here the bioinformatics part in green in a high-throughput sequencing process. So this part is as important as the lab now, but you're well aware about that. we will see a little bit how this different step of bioinformatic analysis can impact your results. If you look on purely data science, the data management means 4 steps: data collecting, data processing, data validation, and data storage. And we will explore a little bit these steps and see also the link, because data processing, it's about analysis of raw reads, the quality control, and so on. This is about identification of the target, whatever it is in your sample. And if you look to data validation, it's the analysis of control. And for example, Natasha gave us some example on the control used in high-throughput sequencing. So let's have a look on these different steps. In terms of data collection, if you look the result of a single sample, in amplicon sequencing, you are in the range of megabase, and in shotgun, you are in the range of gigabase. This means you need to transfer this volume of data to store it. It might be a problem 10 years ago, but now if you look, you can have 1 terabyte backup storage on the cloud with all integrity security measure used by pharmaceutical companies and so on for a couple of €100 per year. So this data storage can be problematic, but not really. We can now store huge quantity of data on the cloud on a secured way, validated by FDA whatsoever, for a few €100 per year. And as mentioned this morning, it was interesting, this exchange is the database also, is the key. And behind that, so storage of the data, but also the database, and as mentioned, the progressive upgrade of the database in terms of allowing the traceability. So collecting data is now easier than it was before, because also the network, the informatic network in our countries is really improving. We are discussing about transferring photos or video, high quality video here, if you make the comparison with our data sets. If you look on data processing, I will introduce the first key message of today. Think bioinformatic triade. It's in the standard, it's in some publication, but it's really important because this triade is in fact the database you use, the algorithm you use to treat your data and to compare your data to the database, and the parameters you have set for your algorithm. These 3 elements can completely bias your results or not. If you want to keep a good accuracy, you need to always think of this triade of elements. Which algorithm, with which parameters, on which database. This is a key message here that we have to take into account. To give you an example of the impact of this triade, it's here an example of a collaborative study. We were more than 20 labs. I just delivered to all of them the same data sets, exactly the same, and they analyze it by their own pipeline. And I won't go to detail, but I want to underline 2 elements. So you have all the lab ID, the sensitivity of detection, and you see the average sensitivity of detection from the same data set was ranging between 35% for lab O to 100% for lab R. So it shows you the huge impact of this bioinformatic triade on the result you can generate. You get plenty of false negative depending on the database, the algorithm, and the parameters that you use. Behind that, if you look at the different steps of the process at bioinformatic level, the first step of bioinformatic level is analysis of raw reads. There, it's a scheme with the different existing steps. And if you look, this bioinformatic triade can pop up or can puzzle your results at each of these steps. So it's important to take into account that. If we look for the second step, which is the identification of targets, that's the same. Bioinformatic triade can impact your result at different places. Whatever the pipeline you use, you decide to make a mapping on reference genome, you decide to make generating context and then comparing the longer sequence database, you have always this difficulty and you have to think about this, which means that validation is primary importance also for the bioinformatic pipeline. We will have a talk on that afterwards. If we look, the third step, which is data validation, and that's been touched with by Natasha before, the key elements is the controls. And we had a very nice example at NIP with the different kind of controls, internal, external, and so on. The key elements visually, that's before high-throughput sequencing, you have positive control and negative control. And in fact, with high-throughput sequencing, a positive control for a target, and I say here viruses because I mainly work on viruses, but for a target is in fact a negative control for any other virus, speaking about external control. And it introduced this second key element, I don't put this image to frighten you, but just to impact you. I try that you keep in mind the fact that running high-throughput sequencing would need an additional and a new control, an external control, which is the alien control. What is it exactly, this alien control? It's in fact a matrix similar to your sample, with all the targets that cannot be present in your sample. So you sequence it in parallel to your sample. And what could you see thanks to this external control? I show you an example of what we did in our lab. So I'm a bit full because I'm just showing you all the contamination we can have in our lab when we do high-throughput sequencing, but I think it's important to share with you all these kind of troubles. My alien control is in fact a wheat sample. So the alien control is here. It's a wheat sample infected by BYDV. And all my sample, all the other samples, they are banana sample. BYDV does not infect banana. What we show is that in all these read numbers are normalized, but in my wheat sample, I get 125,000 sequence of BYDV. Normal is my alien control. And what I can do, thanks to that, I can monitor, in fact, all the contaminating reads that I have in all my banana sample. I give you the worst case, the worst batch that we had. But that means, in fact, and that's also a thing to take into account, I know that I can have a slight background contamination of my alien control in my sample, but it means also that, for example, I have here, let's say, 16,000 sequence of one target of banana. What do I do with this number of 453 reads? So is it a contamination from this sample, or is it a very low level infection? Here we can say, well, it's low level, cool. But this one, for example, 114, it's quite a lot already. And I can have it with my alien control. So this one, I am really in doubt. I cannot say, okay, it's contaminated. Maybe it's in fact another contamination because I have 16,000 here. So that means here, this alien control allows you to really monitor how your contamination burden. And I should give you another example so you go back with a better view. Another batch, I get only very few reads of my BYDV contaminating the other sample. And I can show you the different batches. It's random. This is the same person doing the same protocol in the same lab, and we have different results from one time to another. But also this alien control, I can use it to monitor the contamination with banana targets, banana viruses. And I see, for example, here I have a lot of CMV, but that's because, in fact, CMV is the highest. There is a sample with a very high number of CMV reads. Say that, well, I show you that in terms of transmitting the information, but also because this means also that we can add an additional bioinformatic analysis on these numbers. And that's what we have done. It's currently under publication. We have developed an algorithm allowing to make an automated detection of possible contamination event in order to reduce the burden of decision. It's not the ultimate decision, because at the end, this is the scientific expertise. This is you that will decide it's a contamination or not. But for example, here we add a new tool, which we call it Cont ID, for identification of contamination, which allows to guide you in the decision related to the actual infection or the contamination. After that, data storage. I give you an example here and with numbers on the NovaSeq S4 technology, the highest throughput of Illumina currently. It generates between 2.4 and 2.3 terabytes, sorry, representing 16 to 20 billion reads. That means that if you want to sequence a virus of 10, well, you want to reach 10 million, it's high. We were discussing about 1 million before. It's only 2 or 3 gigabytes for this sample. So again, as I stated at the beginning, data acquisition, it's not a big deal now. We can store it properly. We still have to think which intermediate results we have to keep. That's a key problem, because if you start mapping, you can generate a lot of redundant results. So that means that in addition to cognitive power and an algorithm, the storage capacity will ease your work in high-throughput sequencing. And the last but not the least element is the user-friendly interface. 15 years ago, we were working in this interface. I remember looking for alignment in this white and black screen. I mean, I was young and with a good view, but now, I don't know. But clearly, this was like that. Progressively, we get the second step, second generation, which was the user-friendly interface. Usually, you have to pay for it. We mentioned CLC this morning, there is Genius, the two big competitors. But that's something that facilitates the adoption for us. There is a third evolution in the interface, which is virus-oriented. A lot of colleagues develop, and here I have an example only for viruses, a pipeline fully embedded that you can install on your computer or on a cluster, and you just run it. It's designed to detect viruses, but yet you still have to have a bit of informatic knowledge to install it. And the fourth evolution, which is also very important, and I think it's also in mind of several of you, it's about this HTML-based interface. You just upload your sequence on HTML-based interface, and it delivers you what viruses are present or what targets are present. I showed some example here of directly available tools, because this is also something which will facilitate the adoption for all of us of high-throughput sequencing technologies. So, last very important point, then you are well aware of it, we take into account this in the guidelines, this is about the integrity, safety, and the risk of hacking the data. We have not super financial touchy information, but our results can be touchy sometimes in terms of trades, in relationship between countries. So this is something we have to add in mind. There are solutions, they're a bit more expensive to store the data and so on, but that's something we need to take into account for the whole process, from the collection of the data to the service provider to the storage that we decided to use. This, this has been mentioned by Charlotte, so I don't go more in detail this. We have much more details on all these elements in two publications that also have been transformed and adapted to an EPPO standard, explained by Charlotte this morning. And the key message is this one. We will go progressively, it will be easier for us, and each day will be easier for us to handle this quantity of data, thanks to these three big evolutions and the development of a user-friendly interface. For this afternoon, from the survey, I will launch several questions here. So thanks for filling the survey. We will use it as a starting point to exchange between each other this afternoon. I underlined 4 points related to the setup and the informatic part of the high-throughput sequencing. So these are, thank you for your input. It will serve as a first step for the workshop. Finally, I really want to thank the hundreds of plant pathologists, mycologists, virologists, bacteriologists, nematologists, and even entomologists to the collaboration. Yes, they are not plant pathologists. If you say that to them, they are, I don't know, but really, it has been really a nice collaborative work in the frame of the Valley Test project, but also before in the frame of the cost action, to bring all this reflection progressively. So I would not be able to present anything without all these guys. You may recognize yourself maybe on there. And also, thank you for your attention.
Thank you very much, Sebastian, about taking us in these travels through data in time, and yeah, definitely. Thanks. Well, it's good for you that there have been some positive evolution about this issue of looking at the data out of HTS. Questions? Yes, in the back.
I have a question on these contaminations in the vector that you are developing. Like, this is based on frequencies or on a cutoff level based on these reads that you find across the samples. I think a little bit more if you can share already.
Yeah, well, we have a GitHub page available, so I can send it to you anyway. Okay. But the point is, it's based on the relative frequency. So if you have 1 million reads on a target in a sample, you have a big odds of having a few reads in the other sample. So this is the first element, the relative frequency between the highly contaminated and the others for your alien control and for all your targets. That's the first element. The second element is also related to identity. We look in the supposed contaminated sample if the sequence is 100%, or well, it's not 100%, but it's identical to the contamination source also. That means if it is identical, you cannot yet assert its contamination, but at least if it's not identical, you know it's not from that. So this is based on multiple factor. We tested tens of factors to see what could be reliable on different batches, but it's clearly also related batch per batch. The threshold that is defined by the pipeline is really varying between batches for that, but it's mainly relative frequency and identity.
Just a short question. How to choose your alien control if you are working on the systematic material?
So whatever you do, I exchange, for example, with bacteriologists, which species should I use as alien control? And we found species that are like only present in hot spring. So you know they are not present, they will not be present in your sample. So it's about choosing a target that you're sure it's not present in your sample. If you are on only one plant species, may be easier in there, whereas if you have several plant species, which is easier than if you have just on water. So it's really about thinking on what can I find in my sample and identifying bacterial, fungal, or viral species that is not expected to be there. For example, another show an Andorna virus from Phaserus vulgaris. They're pretty nice because this Andorna is restricted to this species. So it's a really nice alien control, except if you analyze Phaserus vulgaris sample. So it's always thinking, my sample, what cannot be present in my sample? Or you can use tropical restricted organism to control temperate organism. Here.
Well, for half a year. Yeah.
Any other questions? If not, Marcel, you wanted? No. Thank you very much, Sébastien.